Meiosis is a type of cell division that produces four haploid gametes, each with half the number of chromosomes as the parent cell. Through the process, reproductive cells, sperm or egg, are formed. We will focus on the production of sperm cells, or spermatogenesis. The division starts with the interphase of the diploid parent cell, which is identical to interphase in mitosis. In the gap 1 phase of interphase, materials necessary for the division are synthesized. In the synthesis phase, DNA replication duplicates the uncondensed chromosomes inside the nuclear envelope. Lastly, in gap phase 2, the centrosomes in animal cells, each containing two centrioles, are duplicated. From here, the process begins with meiosis 1, or the first meiotic division. Prophase 1 is the longest stage of meiosis. The division begins with the condensation of chromatin, complexes of DNA and protein, into distinct chromosomes, and the formation of spindle made of microtubules between the two centrosomes. Shorter microtubules are called asters. The resulting chromosomes in this process have two identical sister chromatids, and kinetochores form on each chromosome at their centromeres, connecting the sister chromatids. The microtubules are then attached to the kinetochores. Homologous chromosomes loosely pair in the synaptomal complex, a lattice of proteins between the homologous chromosomes, zips up the two homologous chromosomes. From then, crossing over, or synapsis, occurs between the homologs as they exchange part of their chromosomes. The exchange segments are called chiasmata, and the pairs of homologous chromosomes form a tetrad. This process enables the recombination of chromosomes for varying genes. The spindle microtubules begin to extend, and the nucleoli is no longer visible at this point. Around this time, the nuclear envelope fragments as well. By the end of prophase 1, the homologous pairs are attached at their kinetochores to microtubules and have begun to be pulled towards the metaphase plate. In metaphase 1, the tetrad homologs, which contain the two different alleles for each gene, line up on the metaphase plate at the center of the cell. One chromosome from the tetrad is closer to the centrosome on one pole, while the other is closer to the other pole. Each is attached at its kinetochore to a centrosome. This process is the reason behind the law of segregation, which states that each gamete carries only one allele for each gene, and that each allele has an equal chance of being passed to offspring. The law of independent assortment describes the individual separation of alleles interdependent of the others during meiosis. The cell then continues on to anaphase 1, where the kinetochore fibers contract and the non-kinetochore fibers elongate, separating the homologous pairs and pulling the chromosomes towards opposite poles of the cell, centromere first. The structure formed is called the spindle apparatus. Sister chromatids are not separated in this process. In telophase 1, a complete haploid set of chromosomes is at each pole of the cell. In animal cells, a cleavage furrow appears, dividing the parent cell into two daughter cells. In plant cells, a cell plate forms. The separation of the cytoplasm is called cytokinesis. No DNA replication takes place after this stage. Two new cells, each haploid in their number of chromosomes, but with chromosomes having two sister chromatids, are the result of meiosis 1. After the end of meiosis 1, the cell enters prophase 2, which resembles prophase 1. The nuclear envelopes disappear, and the centrosomes duplicate again as a spindle apparatus is formed. Microtubules extend across the cell to connect to the kinetochores of individual chromatids, and the chromosomes begin to move towards the metaphase plate. In metaphase 2, chromatids, which are genetically different from each other, line up with their centromeres on the metaphase plate. The kinetochores of each sister chromatid bind to the microtubules from the centrosomes at opposite poles. During anaphase 2, the sister chromatids are separated at their centromere and move towards opposite poles of the cell. At the end of anaphase 2, telophase 2 occurs, decondensing the chromosomes and reforming a nuclear envelope. Cytokinesis then occurs with the formation of the cleavage furrow. The two cells produced by meiosis 1 divide to form four haploid gametes. The four gamete cells are separate and are genetically distinct. So, by the end of meiosis, an egg or four sperm cells have been formed.